Good morning to everyone and welcome to this event. I think that we can start because the first 3-4 minutes are just for an introduction. So if somebody is late, we will join us later. And so my name is Iris Bertani, and I work in the Cedric project here at the ICGB of Trieste in the bacteriology group under the guidance of Professor Vittorio Venturi. Before starting and give the word to our guest, I will just make a very little introduction, a short introduction about the Cedric project, so you know your host. And the Cedric project is an interreg project that let's say, yes, okay, I can move, that put together 8 partners from Italy and Austria that have the idea of exploiting the microbiome biodiversity of the soil to promote sustainable agriculture. In the framework of this project, we are developing a pilot project that would like to transplant or inoculate, not in the field, not 1 or 2 or few strains, but the entire microbiome. So we are developing a method to make en masse transplantation. And in the same project, we are also developing a platform that join together different actors interested in the agricultural sector, so farmers, scientists, and agrobiotech companies, to share and invent and discuss about methods to translate the scientific knowledge that we have acquired in the labs to methods that are applicable then in the field. And finally, being an interreg project, we are organizing a series of dissemination events to underline and highlight the role of a healthy microbiome in the soil and the importance that this could have in the agriculture. Within this last object, we are organizing this series of webinars. And here it is, our web page, our starting page for the webinars. This will be 10, and we will highlight under different point of view the importance of microbiome from different point of view, like soil health and biodiversity, bioinoculants for agriculture, sustainability, and the communication that has to be done to make this concept of sustainability acceptable and yes, discuss. So now I will just present you our first guest, that is Dr. Mendes. No, sorry, before this is the list of the webinars that are already planned with the dates, but Maria, with who you are in contact, can share this with you later. And so here is our guest for today. Is Dr. Mendes. Dr. Mendes is a researcher. Now I read because I take note about him. Is a researcher at the Universidad Politécnica de Madrid. He has a biochemical and biological background that he constructed during his bachelor and master at the University of Coimbra and during his PhD at the University of Aveiro. And he is an expert in different fields that are related to soil multispecies system, ecotoxicology, environmental pollution, biomarkers, and terrestrial invertebrates. Before giving the floor to him, I just make some technical notes. So please pay attention to be mute, so the background noise will be very little. At the end of the Dr. Mendes presentation, we will have a question and answer time. So if you have any question, raise your hand with the buttons of the program, or write the question in the chat, and then at the end we will make a discussion. And third and last, please remember that the webinar is recording and that you can find the recording in the project website. So now I finish and I leave the floor to Dr. Mendes. I stop sharing my screen and allow him to share. So please, Dr. Mendes.
So, good morning, everyone. Thank you for being here. Thank you, Aris, for the nice presentation and for the opportunity to present this talk on this Cedric webinars and in this case, focusing on soil health and biodiversity. The title of my presentation is Give and Take, the role of Mesofauna and earthworm in soil health. And so, given the scopus of the project, I will talk about the contributions of soil biodiversity to health. Well, the definition of soil health, according to the United States Department of Agriculture website, can be defined as a continuing capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. Throughout this talk, I will give an introduction on current environmental changes, urgent needs, and some steps that have been made forward. Also, we'll introduce the key groups of soil, mesofauna, and earthworms that will be the focus of this talk. And then I will cover 4 topics in which these organisms can contribute to soil health, finish off with the concluding remarks and some acknowledgements. So, carrying on to the title, Give and Take. Why give and take? Well, truth is, we are always giving something up to the soil. We are promoting disturbances, we are giving to the soil chemicals such as pesticides, we are giving it new infrastructures and mold it to our needs, as well as we also are giving to the soil anything that we don't need, like plastics, electronic wastes, and so on and so forth, residues that are released into the soil in massive amounts. On the other hand, we also take. Since many centuries, from the rudimentary agriculture to intensive one, we have been taking up the resources from the soil to feed ourselves. We are also exploring mining resources. And finally, we are also taking land to build up infrastructures and also for the urban development. This habit of take has led us to this tipping point where we have massive areas of degraded soil, a loss in general biodiversity, a change in landscape, higher than ever greenhouse gas emissions, and increasing temperatures. So we are essentially moving forward the doomsday clock to its end. Or are we all doing it? The good news about this is that for the past 20 or so years, though we have an increasing awareness regarding the need to restore, protect soils, and look for more sustainable ways to explore our soils and its consumption. For instance, we now have a group of goals. One of them is actually soil restoration and protection. We also have now the awareness that we need to reduce our consumption habits or level of consumption, but also look at recycling and reusing waste, giving it a new life and repurposing it in a safer way. Also, performing less invasive methods. And this awareness is reflected by the fact that in the past 10 years, the number of publications citing the scientific jargon, let's say, of soil health has increased substantially over the past 10 years. So Could we have reached already the end of this talk? Truth is not. There are more things that meet the eye, especially if we are looking from a microscopic scale from up above. But if we go down into the soil, let's focus ourselves as we would be inside a soil sample. We would look up and we would realize that us humans could be look large, enormous, like some gods that could give and take on our whim to whatever was beneath us, in this case the soil. But we are not gods. So there are many things we cannot control. There are other actors that are in play and much more important. Let's be honest. Let's go back into the soil. If we look closely enough, we can see an array of organisms, very diverse, quite different in terms of abundance as well. And in particular, soil fauna abundance and diversity can interact with the physical chemical properties of the soil and contribute to this thin balance that we so-call soil health. The ways that the soil fauna can contribute to soil health can be organized in four more different areas: bioindicators, toxicological assays, bioremediation tools, and having a protection and support action that will look explained more further down. But to understand this, we need to know the cast of actors that we have selected for display. The soil fauna is quite diverse, um, with different shapes, colors, as big diversity in terms of abundance and also in terms of size. As you can see here, actually some of these organisms can actually be smaller than some of the soil fractions that we normally classify, meaning that they interact quite differently with the soil. During this talk, I will be focusing on soil mesofauna and also a group of macrofauna earthworms. Going more in depth, starting with the soil mesofauna, I would like to talk about three key groups: columella, incatrates, and mites. And why? Well, for starters, they have quite large families and quite abundant in terms of species, and can be easily found across the world. Therefore, they can be a reference. Going into more detail in each one of them, columella, for instance, have an exoskeleton. They're hard bodies. They are essentially detritivore, meaning that they feed on litter that is spread across the soil surface. Therefore, they have a large grazing activity. This activity promotes aeration of the soil surface, meaning more gaseous changes and entry of water as well. On the other hand, we have incatrates. In contrast to columella, they are soft-bodied. These are oligochaetes, small worms, let's say. They have burrowing activity, which leads to the formation of small galleries that are easily colonized by microbes. These species are quite sensitive to pH, salt pH, and they need high moisture content they have for their preferred habitat. Together with columella, they require that soils have high content of organic matter to survive. While these two require that the soil has organic matter, mites can have diverse feeding behavior. Can be predatory, and for instance, they can feed on columella and incatrates, can be herbivory, feeding on plants, and saprophytic, meaning that they feed on dead organic matter. They can lead to changes in soil structure. For instance, by predation, they increase the general grazing of not only themselves but other organisms in the soil, therefore promoting changes in soil structure, which can then promote the microbial turnover. The abundance of mites is quite intertwined with the abundance of other species given its feeding patterns. Another group of soil species that I would like to highlight are earthworms. More easily found, they are visible to the naked eye. They are quite sensitive to the moisture content in soil. They also require high content of organic matter, and they perform bioturbation, leading to the formation of large galleries, changing the soil structure, but also they can ingest soil. They digest the organic matter, they also incorporate inorganic and organic matter, and then excrete this into a cast, promoting the mineralization and aggregation not only of soil particles but also of matter. Given its size and the large quantities of soil it can actually ingest and process, they have then a higher impact than the other species on microbiodiversity and biomass. Now that we have presented the issues and also the key actors, we'll look at their characters, how they act on soil. These four areas that I've selected are bioindicators. They can act also as targets for ecotoxicological assays. They can have an use as bioremediation tools and also promote the protection and support of the soil health. And how? Well, by their role as bioindicators. As I mentioned before, they are quite susceptible to changes in soil properties. Therefore, the changes they are quite frequently used as bioindicators for changes in land use and also other factors, for instance, climate, by correlating their changes in abundance to the changes in properties in the soil. A good example in their use as bioindicators for land use and agricultural management is this paper from Vanoli from 2024, in which they assess the changes in land use and notice that the changes in abundance for columella and mites was mainly due to the soil texture in which these changes were taking place. Other species such as Int'l can also be used in bioindicators, but in this case I will also focus on earthworms. This paper from 2024, they observed that the change from natural grasslands to crop rotation resulted in a loss in abundance, diversity, and biomass of earthworms that was related with a decrease in organic matter and soil humidity. So, therefore, although they are indicators for current land use against new management practice, we can all say that there is, for now, no one fits all solution for this issue. These organisms can also be used as bioindicators for contaminated salts. For instance, for metals, I have here a paper in which they correlated the abundance of arthropods, including columella and mites, to available metal content, and also variations of metal according to the vegetation cover that was found in those areas. Also, earthworms can be used as bioindicators for metals. In this paper, for instance, they observed that the abundance of earthworms would increase as the distance to the source of pollution of metals increased as well, and going from metals to other contaminants such as hydrocarbons. Also, it was observed that the arthropod, columella and mite community was greatly changed with increasing distance to the pollution source. Moving on from their role as bioindicators for both land use and contaminated soils, we have their use as target for ecotoxicological assays. And why? If we look at the concept of soil health, going back to the first slide actually, I would like to focus on a vital living ecosystem. By contrast to this phrase, we have the concept of disease, mortality, and loss. Well, this is mainly due to toxic substances, and based on this ECA inventory, a large number of toxic substances have been identified and have been actually quantified in soil even more, meaning that they can pose a risk not only to organisms, plants, but even to human health. So, before these substances reach the soil, there is the need, as soon as possible, to evaluate toxicity to soil organisms. That is why, for the past 40 years, there has been the development of guidelines to test these chemicals in soil organisms. For instance, these four group of species that I mentioned before, Colambla, Inquitrix, mites, and earthworms, guidelines have been developed to test chemicals using each one of them. These have been applied to many studies across the past 30 years. For instance, for the toxicity of metals, organic compounds, and more recently to contaminants of emergent concern, such as microplastics, nanomaterials, and nanoplastics, for instance. Therefore, this accumulated and incremental knowledge based off the study of these testing these guidelines has allowed regulators such as the EFSA or the European Commission to make informed decisions for soil management. But can we go even beyond current status guidelines? The answer is yes. For instance, in this paper from 2015, by exposing the Columbian species Phosomia candida to silver nitrate on the left and silver nanomaterials on the right, we observed that within the proposed 28 days or time frame for the standard test, we observed toxicity for silver nitrate, but we could barely see, in terms of survival, for instance, any effect on by the nano exposure. However, by performing an assay on these organisms throughout the first 10 days assessing the antioxidant response, we observed that in contrast to the silver nitrate response that was earlier, the response to the nanoform was observed later. This suggests that although within the standard time frame we could see the toxicity of silver nitrate, it is likely that the toxicity of nanomaterials could be seen further down in time. This promising result led us to another study in which we observed that soil biodiversity is full force. In a small mesocosm, we used one oligochaete species, Porcolemblan, and one Cari species, and exposed to copper chloride and copper oxide nanomaterial, not only for 28 days, but down to 84 days, around 3 months, assessing the long-term toxicity. And it was interesting to observe that while there was an immediate effect of high concentration of copper chloride, the nanomaterial form toxicity could be observed later in time and actually could overturn the, the toxicity brought by the chloride. In fact, one key result was that small columnar species, for instance, the Mesofauna macrocata, were highly affected by copper contamination, um, as well as mites. And how could we explain this? Well, truthfully, species interaction can have a role in this. By performing a prototype predator test, we exposed in contaminated soil, we exposed the mites and some juveniles from each one of the four Columbian species with different sizes. We observed that the mites would prefer in contaminated soil to feed on the smaller species, meaning that not only the exposure to a toxicant but also the species interaction could modulate the effects. Other tests designs that go beyond the standard guideline are, for instance, the use of multi-generational tests, in which, in this case, by exposing the initial um, generation of phosphamic candidate juveniles, when they're moving on to adults, to silver nitrate, their reproduction, even when moved into clean soil, was affected at later generations, suggesting that the initial exposure to a contaminant could have impact on the community on the long term. Another way to go beyond what is standard is the use of the less studied, although more organism, Isaiah and Andre, to assess the toxicity of microplastics. Here, you observe that the exposure to high concentrations of compostable plastic bag fragments could have an effect in reproduction in high concentrations. And keeping up with earthworms, I would like to highlight these two papers in which the authors exposed earthworms to endosulfan and bisphenol A, as well as organic UV filters, by complementing what is the standard guideline of the contact test 207 with the gene expression array. This complementation of the effects, the QTech effects, and the effects on a lower biological organization level has the potential to be used as predictor for long-term effects. Also, going beyond the standard guidelines, we can bridge now the gap between controlled laboratory conditions and field and more realistic conditions. For instance, the use of these organisms has been applied in the scenarios of contaminant co-exposure, for instance, the use of pharmaceutical substances and nanomaterials and also metals at the same time, because in real life, let's say, these contaminants are found in mixtures. We observe the effect on survival and reproduction. Also, these organisms have been used when assessing how field soils from close by mining sites could be treated for soil recovery, and how they could be linked to changed soil properties, and also the use of alternative exposure matrix, not only focusing on artificial and standard soils to perform ecotoxicological assays, but also to use or highly enriched organic matter substrates that are more adapt and more suitable to be used for species that require organic matter, such as earthworms. So far, I have covered two of the four aspects in which these organisms can be used in their contribution to soil health. In this case, I will focus more on in more of a passive standpoint, as they are victim to changes in land use, soil properties, and also the introduction of contaminants in soil. But they are much more than victims. They are larger than this, let's say, and have a key role in the development of soil stability. They provide structure, changes in properties as well. They can have a protective role as well, and can be used to maximize benefits from already existing resources, meaning that they are not only passive but also an active agent contributing to soil health. Going into the natural occurring processes, I would like to talk about vermicomposting. As we know, earthworms can feed on soil and other organic matter substances. Once ingested, these go through gut-associated processes (GAP) that involve bacteria and enzymes within their digestive tract. And once excreted, this amalgamation of organic matter can go through cast-associated processes (CAP) related with time, leading to the maturation of vermicompost. This vermicomposting has been studied for its potential benefits, namely in the development and improvement of fertilizers and also in the mitigation of soil pollution. One example of this has been the use of vermicomposting of litter from an invasive tree, Acacia delbata, and comparing the raw material to the vermicomposting composted litter, right, we saw that the vermicomposted litter had less effect in the Lepidium sativum root elongation, meaning that there was no significant effect on their germination, therefore protecting already existing plants. Another example of vermicomposting is regarding its use for the degradation of compostable bags, limiting its potential toxic effects on soils. Also, earthworms have been used in soil bioremediation, namely regarding hydrocarbons. As we can see here in this graph, they can actually have a large impact on across days on the hydrocarbon content in soil. Therefore, the vermicomposting and earthworms can be used as tools for more complex scenarios. For instance, we now know that in agriculture practices, plastic is quite commonly used as silos to store hay, also manure. And at the same time, there is an issue there that antibiotic usage in cattle production is higher than ever, which can promote the onset of antibiotic-resistant genes and also antibiotic-resistant bacteria as well. And in this review, we have observed that we compiled the information that there is the potential for earthworms to mitigate this issue through their vermicomposting. One, they can act directly on plastics that plastic can be used as act as vectors for these antibiotic-resistant genes, bacteria, but also through their digestive process, they can actually provide a selection of microbiota and cut down on the abundance of antibiotic-resistant genes. Another example of less known invertebrate for the degradation of plastics are, for instance, the mealworm Molitor. Carry on with our key actors of soil mesofauna. For instance, colonization species can actually have an effect on glyphosate, leading to its degradation in soil by modulation of the microbial community. Therefore, these four groups of species can act as bioremediation tools by fragmentation, degradation, by ingestion and chemical alteration of substances with toxic potential, and also indirectly by alteration of the soil matrix, namely the modulation of microbial community, therefore contributing to an increase in soil health. Now, moving on to their protective and support agent role. As I mentioned before, these species can interact within the cells by their feeding behavior and other types of interactions, leading to the development of salt food webs. These interactions can provide support and structure to the development of plants, the growth of vegetation, and actually crop production. However, these are not without some issues or dangers. For instance, pests, pathogens, and parasites. In this sense, soil mesofauna can be used as in biological control processes. For instance, um, the grading of columella and the grading of mites can actually interfere with the colonization by fungi, reducing its biomass. And at the same time, their grazing promotes the selection of of certain strains of microbes, leading to a change in the microbial community. Also, nematodes are known to be plant parasitic and therefore an issue for plants. Luckily, both columella and mites can prey on nematodes, therefore uh, protecting the plant. Finally, as columella can also have a saprophytic behavior, they feed on dead organic matter, reducing the risk of necrosis spreading. Also, earthworms, through their vermicomposting processes, can, namely on apple pomace and soil sludge, can greatly decrease the abundance of pathogens so that this organic matter, when reaching the soil, if so, does not have such high risk for, for the system. Similarly, earthworm can actually secrete fluid that has antifungal activity. So there's the potential application for crop protection. In all, both soil mesofauna and earthworms can act as pest and disease control systems. Last but not least, their role in terms of support in nutrient cycling. As we know, these organisms like to live in habitats with high organic matter content and have a diverse feeding behavior. Therefore, throughout these many routes, they can contribute to the transformation, transfers, and fixation of organic matter. In this review paper from 2024, we have some good examples as how they can do this. For instance, springtails and mites can graze on litter and organic matter, changing how these can be aggregated and particulated across the soil, making more or less available to other organisms. Similarly, earthworms can also interfere by ingesting soil and therefore altering how the solid and the solid organic matter ratio is present in soil, and also by its bioturbation, create calories and affect the soil bulk density. Also, once the soil organic matter is excreted in cast, there is also in the cast can occur the fractionation of carbohydrates chain, promoting humidification processes, and also the excreted enzymes can actually promote the alteration of salt pH. Therefore, the availability of nutrients is changed in this way. Not only this, but also these organisms can promote not only the selection but also the transport of microorganisms. They can graze on fungi and select the species that are onset. Through this process of selection of the microbial community that these organisms can perform, also the organic matter processes that are ongoing in soil can also be altered. One curious fact is also that these organisms can also provide some carbon nitrogen input. For instance, columella, their molting process, leading that they release a pupa as they grow up, is a carbon nitrogen input into the soil. Therefore, through direct and indirect processes, these organisms can also contribute to the carbon nitrogen ratio, the bioavailability of nutrients, and also promote the development of structure of the rhizosphere and its stability. So, from their more active role through the bioremediation of contaminants, acting as pest and disease control, modulation of the soil structure, and its involvement in carbonate and other nutrient cycles, these organisms can promote the restoration and maintenance of soil health, being actually ecosystem engineering. So, having covered these four areas, I have moved on then to the concluding remarks. When talking about soil biodiversity, what I presented here and what we know actually is only the tip of the iceberg. In this presentation, I mentioned four groups of soil mesofauna and earthworms and how they could, through four different pillars, contribute to soil health. At the end of this talk, we now realize that the key actors who actually gives and takes to the ecosystem and indirectly to us is the soil biodiversity. For instance, these organisms can give us information for risk assessment. They give up to the soil in terms of transforming organic and inorganic matter. They give the soil structure and stability. On the other hand, they take up and take out of the soil some of the toxic potential of substances, being able to mitigate pollutants as well by taking up some of them and metabolizing them. They take out pests, pathogens, and parasites from the soil, lessen the hazardous potential, and they provide us with a better understanding of soil systems and soil dynamics. Therefore, giving us information for the prediction and decision for sustainable ecosystems, and finally, they contribute by restoring and maintaining soil health. But to do so, there are some key aspects that have to be covered in the future. Obviously, there's a need to protect soil biodiversity. To do so, we need a proper characterization of soil fauna and diverse habitats prior and after man-made activities. We have to take into consideration that every action has a reaction. So, if we plan on restoring soil, maximizing crop production, and even test out and apply new products to the soil, we have to consider that soil biodiversity will always have a key role to play in how the soil system will develop afterwards. Therefore, key and essential to soil health. So, to finish up, some of the papers that I have presented here come from literature, others come from my own experience. I could not have done it without the help of these colleagues and supervisors across my career, and also the contribution of these 5 universities and respective research centers. So, thank you all for your attention, and that's it.
Was really very, very interesting because from my point of view, I always work with microbes, so something completely different. And I'm very happy to see that there are a lot of correlation between mesofauna and microfauna. So it's very interesting. So if somebody has question or can write on the chat or raise the hand. Vittorio, I think.
Well, yeah. Well, thank you very much, Luis. I agree. I echo Iris' comment about we live with microbes all the time, and so it's nice. It's very nice, and I think it's important that we interact more. You know, we are studying microbes, and you're studying, you know, non-microbiological soil members. So no, I just have curiosity. I mean, the first general question I have is how much is NGS, next generation sequencing, used in your studies? I mean, do you use it regularly? That's the first question I have. And is it because in us it's NGS has revolutionized the way we can do things. So I was wondering what has the effect of NGS done to your field? That's the first question. And second, yeah, I mean, again, very general question is, you have mentioned that microbial community can be driven by these soil members. So I was wondering how much is going on between how much interaction are you having with microbiologists in terms of microbial life, or even microbes living within these organisms, the driving of microbial community and these organisms. So how much is this happening in this field? So these are the two general questions I have. Okay, thank you.
I can start by answering the NGS question. Well, it has come and revolutionized. It's more and more frequently used. It has the potential to give us many answers, but it's always a cost. That's the issue, for instance, in terms of using it, but it's more and more used, and it's frequently being used nowadays in earthworms. And this actually leads me to the answer to the second question. How much of these individuals interact, and how can they be important? Sometimes you can make actually the joke that, for instance, earthworms are just simply digestive tubes. They reflect whatever is their microbiota in their digestive tract is a reflection of the microbiota in the soil. So it's a give and take, let's say, of whatever comes in, some stays in, other goes out. And also columella and also incatures are very dependent. For instance, if I can recall correctly, I remember that, for instance, for the strain Fusarium, that columella will always have vector activity with them.
Okay, that's good. So following up from, I don't know, Iris, can I continue? Iris, can I continue? Ask. Okay. Following up from this, in terms of applications and products, what is the market like? What is the application side of things? Are there any products which are successful, or any companies, startup are now trying to use these living organisms in agriculture in order to augment or to monitor or to at least influence the presence of these living organisms?
Yes, I can recall two examples from this. For one project in Spain, in Galicia, that they used actually the litter from vineyards and vermicomposted it and use it as fertilizer for itself. So basically a circular economy in this sense, using the earthworm vermicomposting processed by earthworms. Also, I know of company in Portugal that they actually, it's a startup if not mistaken, that they have been using Bioaccumulators for frass and to use it in small pots, let's say. So it's still a work in progress, but I think they have quite a nice potential, because nowadays we are more and more looking into insects for instance, protein sources and their processes. So there is a lot of potential in here.
Okay, I imagine that's what I mean. There's again a lot of potential to also work with microbiologists to develop products which take into consideration these other living organisms. So okay. Well, thank you. Iris, I'll pass you back the floor, because I see there are other people that are asking questions.
Yes, there is Chiara that raised her hand. So Chiara, can you make your question?
Nice to see you, Chiara. Nice to see you. Hello.
Nice to see you. Nice to see you, Iris and Vittorio, and thank you very much for inviting me to this very interesting talk. Thank you very much
Maybe
Professor.
If you can introduce yourself also.
Yes, for sure, yes. So, Professor Mendes, I am a plant physiologist, in fact, and I'm mostly working on the molecular part of plant physiologist. And With Vittoria and Iris, we had a very nice collaboration, and so we also published together. So I'm very interested to the microbiome part of the story.
And you're
But
from
indeed-
Pisa, right? You're from
Yes,
Pisa.
I'm from Pisa, from Scuola Sant'Anna. My question is related, especially on the part of the earthworms, because I have some contacts with a company here in Tuscany, and also for a project that I currently have on earthworm and plants. And I'm conducting some experiments in order to understand, the effect of vermicomposts on plant. So in fact, I have my results are varying very much, because I have the impression that vermicompost is a general, name for something that can be produced by several different species of earthworms, and also it is varying a lot on the basis of the, starting food of those earthworms. So when we speak about vermicompost, it is indeed something very, general, I would say. Is there any effort in order to, to give to all the community very precise protocol in order to obtain always very sound results in application? This is my first question. And then I have a second question, mostly related to Vittorio's question. As I said, I'm very interested on the plant physiology part, and I wonder whether there is anything published about earthworm as vectors of bacteria that are able to help plants, but in the sense of, for example, phytohormones. Thank you very much.
Thank you, Chiara. I will start with answering the second question. I'm pretty sure someone has done it, but on top of my mind, I can't recall. Regarding the first question, it is an issue. For instance, at Universidad de Vigo, in Spain, they have worked with vermicompost on earthworms and the use of different starting materials, so vermi reactors with different materials, and they have that issue as well, that it's not necessarily the process itself that it's perfect, given that it requires the starting material to be already in some type of condition. If, for instance, I mentioned that there is a high decrease in the pathogen concentration or abundance from Swiss sludge and apple pomace due to vermicomposting. However, that does not mean that it's all clean. And although we have a decrease in pathogens, the metal composition, for instance, or other organic compounds that can actually result from the vermicomposting process can actually be hazardous to plants. So from one way, it's good. From the other way, it's not perfect. So it requires the most complete characterization of both the starting material and what we have at the end. Otherwise, we risk to make the solution worse product, actually.
Okay, so every company has its own protocol to produce a vermicompost that is then a little bit different from the others, is correct?
Yes, nowadays, yes. We don't have it as commercial. Let's say it's not a standard commercialized stamp that is always with the same composition. There will probably be, and it also will depend on actually the company, some issues that they have to be assured. For instance, then as we are talking, if I'm not mistaken, the directive of the European Union for the use of waste, they covered the issue of, for instance, E. coli and Salmonella, if my memory serves me right, and some metals. But there are other metals, for instance, chromium, that is not considered chromium cadmium that is not considered in the directive. So there is some gaps that, in terms of product and selling the product, they can have a label that says safe according to the current legislation, but there are tangible conditions that we still don't have all the full spectrum of information that can support effects.
Okay, thank you very much.
Thank you, Dr. Mendes. We have some question also in the chat. There is Daniel that is asking, I read for him because it's a short question, so I can read. What changes could be observed in the microbiologic community associated with earthworm population changes? So if you have an idea of the group of microbes, perhaps, that earthworms likes to eat or likes to make survive in the soil and avoid them.
I may risk myself into getting into a corner that was not good, but I recall proteobacteria and actinobacteria, for instance, are promoted once they go through the digestive tract of the earthworms. The cast actually has proteobacteria and actinobacteria, so they perform some selection on this type of bacteria regarding others. I recall that Fusarium, for instance, the strain is also limited by earthworm activity. So I can say this at least.
Okay. And then
one, actually, I'm reading the question. I don't know if the question actually is changes in population of earthworms, or meaning that by increasing the number of earthworms, this, of course, the microbial community will be changed more quickly, actually. So.
And I think we have to consider this also when we use bioinoculants, microbial bioinoculants, because perhaps they can really affect the result of the bioinoculation if they act on the microbial population. This is something, okay. There is a hand that is raised, but first I have a question from Unarin that is very long. So I think that if Unarin can take the word and unmute her phone, or his phone, his microphone, perhaps he can make the question by himself.
Hello, ma'am. How are you?
Okay. Hi. Please make your question.
Thank you again. I'm Munarine, so I want to ask what
Onarin, excuse me. Please introduce yourself. It would be nice to know who you are and where you're from and what you do. Okay, thank you.
So I'm sorry about that. So my name is Unarine. I'm an honors student from South Africa. I'm working on how we can elicitate a heightened a plant immune response using botanical extracts and some stressors such as extracellular DNA. So that's for my honors. Hopefully, I can also grow it into an MSc next year, a master's. But my issue or question is that what are some of the practical ways you could recommend us on how we can improve or promote soil mesofauna other than no-till? Because normally the first answer to that is no-till, which makes sense, but in Africa and most global south our nations, our no-till is not yet a feasible solution on a large scale, because we simply don't have one advanced our knowledge in the skill, especially amongst farmers, and secondly, we don't have enough no-till or technology. So what other practical methods would you advise? Thank you, sir.
Thank you for your question. I think there is no clear response to that, because for starters, each soil it's its own ecosystem, completely different from another. So soil properties for starters have a big role in how the soil community, being it microbial, mesofauna, or microfauna, develops. So for starters, it requires a characterization of what are the soil properties. And it's not like we can completely control the soil mesofauna that develops within an area according to our management practices. Of course, we are going to benefit one or the other, but at the end of the day, I think it's one by one a situation that it requires more studies, of course, in terms of characterization, what there is beforehand, and therefore what can be done so far. I hope I could answer your question.
All right, so what I'm getting from you is that there's a research gap in that as to how we can transition from our theoretical understanding into practical
Yes,
application.
yes, there's still ways to go.
All right, and thank you, sir.
Okay, now is Marco Contin. Dr. Marco Contin can.
Yeah, thank you very much. I'm Marco from University of Udine, Italy. Thank you, Luis, for excellent presentation. I was just curious about the chemical environment inside of the gut of collembola, earthworms, and mites. I mean, regarding pH, for example, or redox potential. Are these similar because they can play a role for the selection of microorganisms and a role also for solubility, bioavailability of organic xenobiotics? And what is your idea about this environment, chemical environment inside of this gut?
As I mentioned before, in terms of microbiota, they are a reflection of the soil that they can ingest for starters. So earthworms, for instance, can ingest large quantities of soil compared to collembola, mites, and incatrices due to their size. And this can then have an effect on the pH. The pH, the acidic pH and the redox potential of the collembola, if not mistaken, will lead to obviously processes that, compared to earthworms, are less studied, but are likely to be completely different. Therefore, for instance, the bioavailability, as you mentioned, of pesticides will be completely different from species to species. Even the way that they interact, for instance, collembola do not like. They have actually on their legs hydrophobic structures that make them be on the surface of water, but so they are not so much hydrophilic. Let's say, so the availability of one xenobiotic will also depend on how they get in, and actually how they avoid this area before ingesting, whether they can be in the presence of a certain xenobiotic, and then if they ingest it, how can it be affected? And actually, the point, how can it be present? For instance, if a xenobiotic becomes accumulated in litter compared to the soil, it will be completely different the way that it interacts with the organism and it can be degraded within the digestive tract.
Yeah.
So
Okay. Yes.
there
Yes.
are
Thank you very
lots
much.
of things
Yes.
that the completely line of research that can be done there. So those are things still to do.
Yeah. Okay. Thank you very much.
So I don't see any other hands up, so I would like to thank you, Dr. Mendes, because it was really, really interesting. I would say thank you to the IT service of ICGB that helped us to organize all the webinar. And Vittorio, do you want to say something, or I give the appointment to the next webinar?
Yeah, well, thank you very much, Iris, for organizing this, and thank you very much, Luis.
Maria role.
Maria, Luis, and thank you, Luis, for giving us the time and informing us on this, aspect that for us microbiologists that I think we need to interact more as microbiologists and microbiologists with respect to soil life. So thank you very much.
So thank you again.
Thank you.
And I give you appointment to all of you. I would like to see you again on the 29th of January for the next webinar. That was the host is Professor Lucrezia Lamastra, and she will speak about sustainability in agriculture, cultivating a greener future. So thank you again to everybody, and see you on the 29th.
Thank you. Bye-bye.
Okay. Bye-bye.
Bye. Thank you.
Bye.
Bye bye.